should be recognizable as the three-step sequence. So we've got the three-step sequence, we've got the condensation step, it's going to end up releasing the CO2, so that we have 2 plus 3 goes to 4 plus 1, that's Ks. And then the other 3 are just carrying groups and helping them transfer around. And that's necessary just because we've got this big enzyme, and we're taking stuff in and we're building stuff up, and so we have to move things between subunits in order just to enable that whole process to happen.
Which 3 did you say were for the 3 steps? Like steps 2, 3, and 4? The last 3 on that list?
These last 3, reductase, dehydratase, reductase, is NADPH here, NADPH here, because they're redox enzymes.
Yeah.
And then this is the one that's removing the water.
Okay. Thank you.
All right. So this should let me help clarify it a bit. So first step, the AT activity. Second step, the MT activity. And as I said, it could just be called an MAT activity. So we're attaching the acetyl group onto the Ks subunit by virtue of this AT activity. And then the next step is to transfer the malonyl group from Malonyl-CoA onto this ATP using the MT activity. So here's another look at it. We can look at the whole structure of the enzyme like this with the different subunits. So acetyl-CoA comes on, gets added onto the Ks, and then Malonyl-CoA comes on and gets added on to the ACP. So now we've got Malonyl-CoA on the ACP and acetyl-CoA on the Ks. So we just loaded it up, right? So then we're ready to go. We have a condensation reaction that splits off the CO2. Right? So that's the activity of this Ks enzyme. So that generates the beta-ketobutyrol ACP. So this basically is the 4-carbon unit. It's on ACP, right? And that's where reduction, dehydration, and reduction occurs when it's attached onto ACP. So now we're on ACP, but if you look back here, the malonyl group that comes on has to come on to ACP. And we can't do another round unless the ACP is cleared, right? So where does this 4-carbon group go? Right? It goes back to where the 2-carbon group originally went. So that now when malonyl comes in, right, so it goes from here to where the 2-carbon group was on Ks, so there's the 2-carbon group, but now it's a 4-carbon group, right, which frees up this guy, right? So this guy moved away, so now he's free to take another one of these. Does that make sense?
Yeah.
So now you're ready for another round. The only difference is now this is 4 instead of 2. And then you just keep going. Okay.
So a malonyl gets added each time basically
Correct.
to the chain.
It gets added every time. It gets added to this Ks. This ACP every time, and then there's a Ks, there's a group next to it, either 2 or 4 or 6 or 8 or whatever it is, and then the fundamental reaction is the splitting off of CO2 to form this new carbon-carbon bond in here. Yep. So that's pretty much it. That's the secret of the fatty acid synthase. So, acetyl-CoA, malonyl-CoA, condensation, 3 steps, right? Translocation back, and then another one's ready to add. It's not that bad. So, if you just write them down on a piece of paper, just as a list of things, right? One thing that makes it easier is that this 3-step sequence you can just conceptualize as one thing because you already know about it from the other pathways. Okay. So, this is a few slides to basically kind of take you through the details. Questions? Here's the overall reaction. If you start from 8 acetyl-CoAs, you need 7 ATPs because you're making the malonates, right? Notice that this is the overall reaction for both acetyl-CoA carboxylase and fatty acid synthase. Right? So the first enzyme makes Malonyl-CoA, the second enzyme uses it, so there's no Malonyl-CoA in this. Right? Instead, it's all acetyl-CoAs. 8 of those, right, will make a C16 fatty acid from the 8 acetates, and then the CoA is released. Right? You need to do that 7 times. Right? So you go 4, make 4 on the first one, and 6 more to get you to 16. So every round these 2 NADPHs, right? So you have 14, 14, 14. 7 ATPs, which was used in the first enzyme, not the second one, but to make the Malonyl-CoA in the first place, you needed an ATP. Right? So ATP, ADP, PI. And then it generates only 6 waters. Why is that? It should generate a water every time if you're going from a group that has a keto group to a methylene.
The last
Sorry?
one.
You have to use one.
Where?
When, at the end of the
At the end, yeah,
synthesis.
to hydrolyze it off, right. So instead of 7, it's 6. Well, it says right there. Question?
I just read that the CO2 is taken up by production of Malonyl-CoA.
You're whispering.
Oh, I'm sorry. I was gonna ask what about CO2, but then I just read note that CO2 is taken up in the production of Malonyl-CoA.
Right, so there's no net use of CO2, right? We take it up, we let it out, and it's the same molecule, the same carbon atom, right?
Yeah.
It seems like it's energetically favorable to make like even numbered fatty acids and break them down, right? So, or like it's at least in the breakdown. So why do we end up with odd numbered fatty acids?
Because they're useful in metabolism and in cell and in membrane structure. You're right, they're not so necessarily useful for fuel directly, but the cell still has to be able to process them and handle them.
And cells like try like break down specifically even numbered over odd, like target those.
I don't think so.
Okay.
I don't think there's really a capacity to selectively mobilize only the even ones from some, say, a fat cell. And certainly the diet that comes in as a mixture.
Do we synthesize all, like, even, odd, of all types?
Yeah, I'm going to show you how we make the odd ones. This is only how you make the even ones, right?
Yeah.
Not to mention it's only how you make the even saturated ones. How do you make the odd unsaturated ones? Okay, so this was in the cytosol, but right, so in animal cells, in the cytosol, NADPH production, we'll see that there's another enzyme called malic enzyme. NADPH is high. And here we're doing fatty acid synthesis, as we just studied, right? And some early stages of isoprenoid synthesis and sterol synthesis, which I'm going to try to rapidly tell you about. And then in the mitochondria, we have fatty acid oxidation, as you know, acetyl-CoA production, as you know, synthesis of ketone bodies, which are things like acetoacetate and acetone in the liver that get transported through the body to get used as fuel. And then the fatty acid elongation system, right, in the mitochondria of animal and yeast cells. In the ER, we have some elongation, but we also have desaturation, right, which means the addition of the double bonds. And then plant cells kind of mess things up. They put things in different places, but don't worry about the plants. Okay. So remember that this is the acetyl-CoA carboxylase that is using up a carbonate and making Malonyl-CoA. Okay. From acetyl-CoA. But we need the acetyl-CoA for everything, right? We have acetyl-CoA in that enzyme, we have acetyl-CoA coming in and fatty acid synthase. So we have to make the acetyl-CoA available, right? And so here's another one of those transport slides, right, which I know is everybody's favorite. Mitochondrial matrix, cytosol, fatty acid synthesis is going on here. We need acetyl-CoA to do that, right? So here we are in the mitochondria, right? Here at citric acid cycle reactions, malate, oxaloacetate, citrate. Pyruvate can go to oxaloacetate. We know that. That's like a gluconeogenesis step as well. Right? When there's a lot of energy going on, right? When there's a lot of citrate, right? And this makes sense, right? Because that's also when we want to take some of the excess energy and use it as and use it to build up fuel molecules or storage molecules, right? The citrate would come into the cytosol, right? So there's a citrate transporter, and then there's a special enzyme called citrate lyase, which splits the citrate into oxaloacetate plus acetyl-CoA. And so this is the source of this is an acetyl-CoA pipeline into the cytosol so that you can make fatty acids. Okay? And then in the regeneration process, we have oxaloacetate. We've seen this before. NADH goes to NAD, right, so that we can reduce the malate, reduce oxaloacetate to malate. This we saw in the process of gluconeogenesis, right, in replenishing NAD for glycolysis. But so malate can go through because we know there's a transporter for that. But there's another enzyme called malic enzyme. It's a cytoplasmic enzyme. It converts malate to pyruvate, splits off CO2, but this is an oxidation, so we get to reduce NADP and make NADPH, which of course we also need for fatty acid synthesis. So the process of moving citrate out generates metabolites and activates this other enzyme, so we get the NADPH as well. So you should remember that the pentose phosphate pathway, of course, generates NADPH, but when we activate this process of fatty acid synthesis and we bring citrate in the process of recycling, we generate more NADPH. So now we've got another source for it. Questions?
Will you remind me again, citrate inhibits pyruvate kinase, or is it hexokinase?
Citrate activates or inhibits one of the glycolytic enzymes.
Okay.
I don't remember.